This video is going to look at the blood typing lab, and we're going to look specifically at the forward, reverse, and week D testing. Please do not watch this until you have at least watched the video on just ABO groups and RH types in general, because it's not going to make any sense if you don't do that. So just make sure you're comfortable with that information before continuing on here. We'll start by talking about the forward grouping test. Forward grouping in the lab when it comes to blood typing is where you mix the patient's cells that are unknown, we don't know what kind they are, with an antibody that we bought from a company. So in this little image here, you can see some red cells in a tube. Basically, you'd be adding anti-A, anti-B, and then I'll show you anti-D to the tubes. You'll centrifuge them, and you'll look for a reaction. What we're doing here is we are trying to figure out what the heck proteins are on these red cells. And the only way we can do that is by mixing them with known antibodies. So anti-A is a protein that attacks A red cells. Anti-B is a protein that attacks B red cells. And D is a protein that attacks cells that have the D antigen on them. We are using these known antibodies to figure out what antigens are on the cells. The anti-A, B, and D all come from a commercial product. We have removed any plasma or any serum from the red cells that are in the tube, so it's just cells and antibodies. Let's say you put these things together in a tube and you get these results. You get clumping or agglutination, which is the clumping of red cells due to antibodies, in the anti-A and the anti-D tube. Do you know what blood type that is? Pause for a second and see if you can figure it out. If you said A positive, you're correct. So this is because when I added anti-A to the red cells, they clumped. That means that this antibody attacked something, and since I know that anti-A attacks A cells, these must be A cells. It did not attack when I added anti-B. There must not be the B antigen on there. It did attack when I added anti-D. They must have that positive RhD antigen present. This is great, but in blood typing, we cannot stop here. We have to confirm, confirm, confirm. And that's what we do with the reverse grouping. For reverse grouping, we only do this for type A and B cells, for the ABO blood type group. We don't do this for RH, and we'll talk about why that's the case at some other point. But we flip it for the reverse grouping. In this case, the reagent is the cells. So we actually purchase known type A cells and known type B cells. And then we add those cells to separate tubes that have the patient's serum or plasma in it. And in the serum are going to be the antibodies that that patient has. So remember Landsteiner's rule, you have the antibody for the antigen that you lack. And remember that our patient was an A positive patient. So if they have the A antigen, they should contain anti-B antibodies in their serum. The little green snowflakes on the bottom are supposed to represent antibodies. And if we take their serum, we add the reagent cells to it, what would happen, what would you predict to happen in a patient who is type A? They have anti-B antibodies in their serum. I'm adding A cells to one tube. I'm adding B cells to another tube. I add patient serum. You would expect clumping to happen in the B cells because the anti-B that is present in the patient cells will react with the B antigen on the reagent B cells. If your brain is already swimming, pause. Take a break. Rewatch, reread. It gets complicated. It doesn't get easier as we continue. This is just what it looks like when we actually do it on a slide. So the top slide is showing someone who's O negative, meaning none of the antibodies reacted with anything on the cells because the cells don't have any antigens to react to. Very homogeneous mixture, no clumping, no light parts, dark parts. The middle one would be our patient here. So you've got an A positive, you've got clumping because the antibodies, the anti-A reacted with the A cells. Then you have nothing happening for B, and then you have clumping for D. And notice that it's a little bit finer for anti-D, and we'll talk about why that's the case. If you had a B positive patient, it would look like what you've got on the bottom there. this is what it would look like in the tube, and this is kind of what we're going to be doing in the lab, is adding the anti-sera right to the blood in a tube and then spinning it. Big old clump for the A tube, no clumping for the B tube, and fine particulate clumping for the RH tube. This is just kind of showing you the various degrees of reaction. All of these are positive. So all of these indicate that there has been a reaction. Sometimes when we do the reverse typing, you will see hemolysis instead of clumping. So just make sure that you know how to look for hemolysis, you know, that kind of clear red instead of hazy red. And that's because when we use patient serum, we don't know what kind of antibodies are in there. Are they hemolysins? Are they going to actually lyse the cells, or are they going to clump the cells? Either one is a positive reaction. So we want to make sure that we know that for the reverse grouping, hemolysis is positive. This is also showing that sometimes you have to look under a microscope to see it. It can get really super fine. We're not going to do that, but that is something that can happen. Also, when you compare the antibodies for the ABO group and the RH groups, the ABO groups are big. They're cold reacting, so we do our tests at room temperature. They are made of a structure called IgM, which is a particular class of antibody, and they are big binders. They clump real big. Strong agglutination. RH antibodies are warm reacting. They need to be incubated, they need to sit for a while, and the clumping is not super strong, so we have to look a little bit closer for that. The type of antibody that makes up RH antibodies is called IgG, and because of its structure, it's a little different, and it's going to cause a weaker form of agglutination. Sometimes you'll do this whole test, and you will get a negative in the RH tube. And that's fine, that could be truly negative, but there's a variant of the D antigen called weak D. The patient will originally type as though they were Rh negative, but they actually have kind of a weaker version of the D antigen. When it comes to donating blood, we would consider this patient Rh positive. So we have to confirm that all of our negatives are actually truly negative and not just weak D. What happens here, the reason why you don't see the clumping, is because, as I said before, the anti-Rh antibodies are kind of weaker. And when you add the anti-Rh to the red cells, what happens is the antibodies coat the red cells, but they do not agglutinate or clump the red cells. So the little purple Ys are the antibodies, and obviously the circles are the red cells. And what you're seeing here is something called sensitization, coating with an antibody without clumping. This is a positive reaction, but it's not something we can see with the naked eye. It's not even something we can see microscopically. So we have to find a way to make this visible. If this is happening, if we are adding anti-D to the red cells and it's coating them, that's a positive reaction. So we have to find a way to actually visualize that. And the way we visualize it is by kind of adding a couple more layers of antibodies to this. It gets messy, hang with me. We test for the presence of sensitized red blood cells using what's known as the Coombs test. The Coombs reagent is something called Antihuman globulin. Basically, it's an antibody to an antibody. What happens is, if you have these little red cells coated with antibodies, you add another antibody that attacks the antibodies coating the red cells. So that actually attacks like the little handles on the Y. This is AHG, antihuman globulin. When you do that, that will attack the antibody on the red cells, which then will cause clumping. So you have to put one more layer of antibodies in order to see this thing. Normally it's too fine to see, it doesn't cause that big clump formation. You add one more layer of antibodies, an antibody that attacks a human antibody, and then we get clumping. Or if there is clumping, I should say, that means that this patient is weak D positive. And ultimately, RhD positive. If you do this step and you see clumping after you add your Coombs reagent or AHG, they're the same thing, you can stop. This means that, oh, yep, our patient had that weak D form, they truly are Rh positive, we're done. However, if they are negative after we add the AHG, we have to confirm this. Again, it's always confirmation when it comes to blood typing. Gotta be safe, gotta be right. What we're gonna add are something called Coombs control cells. And these are actually reagent cells that are red cells coated with IgG, just like it would be in a patient who had weak D. They will clump when you mix them with antihuman globulin. Coombs control cells are controls for antihuman globulin. Number one, if you mix AHG with Coombs control cells, it should clump. If you do this test and it doesn't clump, it probably means something's wrong with your Coombs reagent or your AHG. They're the same thing, remember. The other thing is, a lot of times these reagents are clear, and it's really hard to tell if you actually added them to your tube. So adding the Coombs control cells and knowing that they should clump because you have the Coombs reagent, the AHG in there, is a confirmation that yes, in fact, you added the AHG. Clumping is good when it comes to these cells. That means we've done everything right. Let's walk through the two scenarios that can happen. On the top, we have a patient who is weak D positive. These are the red cells with that D antigen. And then we have someone who is negative. We're going to add our anti-RH. Add it to both. Notice how those that have the D antigen on it will bind to the anti-D. That's what anti-D does. It binds to D. If you look at the bottom one, there's nothing to be bound to because this patient is Rh negative. We do a wash step, so you kind of rinse the cells off with saline. And what will happen is any antibody that was not bound is going to be washed away. So this is what will be left in our tube. You're going to have some red cells that are coated with antibody, but we can't see it, remember, because it's too fine. It's too small to see with the naked eye. And then on the bottom for our negative patient, all the antibodies were washed away because they didn't ever bind to anything. Now we're going to add our AHG. So remember, the tube looks like it's negative right now. We're confirming that. Add your antihuman globulin. In the top tube with the weak D and the antibodies bound, the antihuman globulin will bind to the antibodies and it will cause clumping. You'll spin it, you'll look, you see the antibodies, or you see the clumping, excuse me. Cool, great, this is actually a weak D patient. No more, we're done. If you add the AHG, you spin it, you look, and it's still negative, we gotta confirm our AHG worked. So then nothing happened here because there was no antibodies to bind. You add your Coombs control cells. Again, they're basically a very similar version of what you see at the top. Pre-made cells coated with antibodies, you add them. The AHG that you previously added will bind to them, and it's going to cause some clumping. Now, this clumping is not going to be big and obvious. It's going to be pretty weak because half the cells you have in the tube are Rh negative, so they're not clumping. But the other half, the Coombs control cells, are positive, and so they will cause clumping. So you have what's called a mixed population, and as a result, you're not going to get a big button or like a big clump. It's going to be pretty fine. You have to look really closely. This is a complicated thing. Okay, this is a super complicated thing. If your brain is like, I have no idea what's going on, talk it over out loud. Let me know how I can help. It's going to be important that you have a solid understanding of this. And if it's not right now, that's okay, because we're going to move into this topic in class so we can really discuss it. But please note that if you're feeling like, what is going on here? It's worth stopping, reading, asking questions. Now, when you come into the lab, remember also when it comes to any sort of reagent, you have to make sure that our reagents are good. We have to make sure they are, working properly. It's just like your analysis. When you get there, typically what will happen is you look at your product, you make sure that it's within date. Now, ours might not be within date, but in a real lab, it needs to be. If there's a package insert, like if you get a box that has a new bottle of anti-sera in it, read it. What's the proper storage? Are there any other reagents that it needs to go with? What is the correct procedure for usage? How do you use it? How do you handle the specimen with this reagent? When you use the product, make sure that you do not mix the droppers. You can't mix an anti-A with an anti-B because that's going to make things messy, right? You're going to be screwing up your results. Don't touch the droppers to the slides or the tubes. That introduces contamination. Add the reagents in the orders specified. And make sure your glassware is clean. These are just a couple ways to ensure that we are getting the right results. Because again, blood banking is really a life or death kind of thing. Your analysis, whatever, you read your leukocytes a little bit off, it's probably not life or death. Blood banking can be. If you have questions, please ask. We will talk about this again, both in class and in lab. I am here to help. Let me know what I can do.